Replication is the first process of DNA, which makes DNA into more DNA. One strand of DNA, called the parental DNA, begins the process. It is unzipped by an enzyme called helicase. On each sides of the two new strands, binding proteins facilitate the binding of nucleotides. There are two strands in replication. The leading strand moves with the helicase in a 5' to 3' direction. Thus, with the help of DNA polymerase III, it creates a long strand of new DNA. The other strand is much more disjointed. It is called the lagging strand. Fragments are bound to the DNA by polymerase III. These fragments are called Okazaki fragments. RNA primers also hold places for the fragments, and they are held in place themselves by primase. DNA polymerase I proofreads the strand as it is created to make sure there are no mistakes. And DNA ligase seals the fragments together. Both strands operate in a 5' to 3' direction. Transcription is the next process, and it makes DNA into RNA. For this process, the DNA strand is not fully unzipped. Instead, RNA polymerase unzips it at the front and rezips it back together at the back. Again, there are two strands. There is the sense strand and the antisense strand. A chain of messenger RNA is built against the antisense strand. It is made in a 5' to 3' direction. As in replication, 3 nucleotides are bound together to make the new strand. This new mRNA molecule can now go outside the nuclear membrane. Translation is the last process and turns RNA into proteins. The mRNA strand created in transcription moves out of the nucleus. Then, a small ribosomal subunit attaches to the start codon. An anticodon of tRNA, with an amino acid attached, comes to rest there. It is surrounded by the large ribosomal subunit. This unit has 3 binding sites within it. The ribosome then moves down the strand of mRNA. It allows tRNA anticodons to move through it. This creates a long chain of polypeptides, otherwise known as a protein. This process also occurs in a 5' to 3' direction.